Hi, everybody. I'm gonna wait like 2 more minutes for more people to show up, just in case. But before that, I do want to kind of discuss how to go about studying for this lecture. I know it seems like a lot and it was very overwhelming, but when you like write it all out into one page by going like slide by slide and putting all of the steps together, it's really not that much. So I have an example of that. This is my notes that I've already posted. It's for the pyrimidine metabolism. I kind of like put everything together from the slides into one page. And as you can see, it's like not that bad. Um, and a lot of these enzymes are lower yield, but I just like to memorize as many enzymes as I can. So, I'm not missing anything for the test. But for Dr. Patel, last year she was pretty straightforward with the questions for this exam. This was our lecture 9 last year, and it was the lecture right before the test. So we had 5 days to study for the test and have to learn all of these pathways. So she kind of gave us hints on what she wanted us to know and what she didn't really want us to know. I don't know if she's going to give you as straightforward answers or questions this year, since you guys have more time to study for it, but hopefully that's the case, in which case I would definitely focus more so on the high yield enzymes, which I will point out as we go along. All right. I've posted these slides already on Canvas, so you're welcome to look at those. And I've also posted my study sheets, which I just showed you an example of. All right, so we'll start now. Okay, so you definitely have to memorize the purines and the pyrimidians, like which are which. So the way I remembered these is the phrase pure as gold. So purine, and then the A for adenine and the G for guanine. And then X marks the spot. You think of gold, you think pirates, X marks the spot. And you get your hypoxanthine, which has an X in it, and then your xanthine, which also has an X in it. For the pyrimidines, the way I remembered them is the phrase cut the pie. So C-U-T, and then pyrimidines for pie. For arotic acid, you just kind of have to remember that because I don't have like a fun way of remembering it. So understanding that these are all nitrogen-containing bases. Sorry, I forgot to put it on slideshow. Okay, understanding that these are nitrogen-containing bases is important as well, and that's what makes them similar to each other. Okay, for this next slide, she's just kind of showing you these nucleosides. So you know they're nucleosides because they don't have the phosphate group. They have their ribose sugar added, and then they have the base added here. The difference between ribonucleosides and deoxyribonucleosides is the presence of this hydroxyl group here on your second carbon. And you won't see that in the deoxy. It'll be missing the hydroxyl group for all of these. So understand these abbreviations because she does like to use them for the deoxygenated ones. But other than that, don't worry about memorizing the structure for any of these. That's too much information. Remember the names, understand what they're made up of. It's just the base and the ribose, and understand the difference between deoxy and the regular ribonucleoside. Okay, so in case anybody was confused as to the difference between a nucleoside and a nucleotide, I have added right here kind of a way to remember that. So your nucleoside is going to be composed of your sugar group and your base together. So a base by itself is just known as a free base, and together with a ribose is a nucleoside. Then when you add a nucleoside with a monophosphate, a diphosphate, or a triphosphate, if there's a phosphate group here, that's how you know it's a nucleotide. So this is just the example that she was showing you to give you the different parts that are important for us to understand. But you don't have to memorize the structure or anything. This slide, she was just kind of introducing concepts that we're going to learn more about as the course goes on. I underlined GTP because it's kind of important to understand that we use this for energy when you're making purines and pyrimidines, as well as ATP because we use it in synthesis of purines and pyrimidines. But other than that, we're going to learn like all of this later on. So it's not really super important to like memorize this because it's just like an introductory slide. And this too, I wouldn't memorize this. I highlighted the coenzymes because she mentioned it, but honestly, like it's just to get you acquainted with the importance of nucleotides. This, however, is a little bit more important to understand. So that what they're talking about is where purines are made and broken down. So purines are specifically made de novo in the liver, and in the liver cells, this will happen in the cytoplasm of the liver cells. Pyrimidines, on the other hand, will happen in liver and other tissues, and specifically within the cells of the liver and the other tissues, you'll see this happening in the cytoplasm and the mitochondria. So know the difference between the metabolism of pyrimidine and purines and the respective tissues, organs, and cell locations. Okay, so we're going to start with the metabolism of purines. She wants you to understand that you have to know the catabolic and anabolic processes. So the biosynthesis has two pathways that we have to know. You have your de novo pathway, and then you've got your salvage pathway. So your de novo pathway is essentially making the nucleotides from scratch. The salvage pathway is really just recycling free bases and then putting them on the ribose and phosphorylating them. And you're not essentially making them from scratch. This is just an overview, so don't worry about memorizing that, but you're going to have to kind of know it in a little bit more detail regardless. So, but the main point of this slide was to introduce the fact that your de novo purine synthesis happens in the liver, as we already discussed, specifically in the cytoplasm, and that your salvage pathway happens in extrahepatic tissues, which is just a fancy way of saying tissues outside of the liver, so not the liver. So this is a super important slide, and knowing most of the details in this slide is important for the test. So you get your ribose 5-phosphate, you have to know this, from the pentose 5-phosphate pathway, which we haven't learned about yet, but we will probably next block. So just kind of associate these two. And since they have similar names, they're kind of easy to remember that one comes from the other. So you have your ribose 5-phosphate, and then with ATP, know that ATP is important for this, and the enzyme PRP synthetase. And she personally likes abbreviations, so maybe memorize the abbreviations, but I'm pretty sure on the test she'll give you both the full name and the abbreviation. So you don't have to worry about that. But sometimes it's easier to remember the full name because it tells you what it's making. So PRP synthetase is making PRPP. So in this case, you just have you have to memorize your enzyme, you have to memorize the fact that it has ATP, or it needs ATP to be made, and memorizing the substrate and the product names. Don't worry about the structure, that's not important. So what's going on here is that you're just adding 2 phosphate groups to the ribose 5-phosphate and then turning it into PRPP, which is a super important molecule for both pyrimidine and purine metabolism. And it'll come back when we talk about the pyrimidines. Okay, this is also an important slide because she likes her commitment steps, and knowing the details of the commitment steps and the first step are important. So knowing this enzyme for sure is what catalyzes the commitment step. So the commitment step is essentially the step in the pathway where when this happens, there's no going back. It's going all the way to the end to make products, and it's just kind of sealed its fate, essentially. So definitely know that you take the PRPP, which we made in step 1, and you're going to add glutamine to it. And then through glutamine, PRPP, aminotransferase, definitely remember this enzyme name. It's going to take the amide group from the glutamine and add it right here to the PRPP. And whenever you take the amino group from the glutamine side chain, it's going to release glutamate because they have similar structures. They just have the difference of the amino group, essentially. And so you add the amino group to make PRA. So know the PRA, know that glutamate is made, know that know the name of the enzyme, and then know the two substrates that are being reacted upon. Lucky for us, we don't have to know any of these steps. I would just kind of suffice this to say that there's multiple steps after PRA that ultimately allows you to make IMP. I kind of highlighted these so you kind of get a general idea as to what goes into these multiple steps. But I probably wouldn't memorize them. I just kind of recognize them, I guess. So ultimately, when you make IMP, IMP can now either make AMP or GMP. So here is how IMP makes GMP or AMP. So following those multiple steps that we discussed, sorry, there's a question in the chat. Should we know how many are ATP dependent and or involve amino acids? So when I studied for this, I did remember that just in case. I'm pretty sure her questions, I mean, you're going to get 6 questions, 5 to 6 questions per lecture. So, I mean, I'm sure she's going to focus on the big picture ideas. So my best advice is to have a general idea as to which ones use ATP, but go first and go through and memorize the main enzymes that she wants you to know. And then if you have more time when you're studying, to go back and memorize the details. However, for this particular slide that I'm on right now, you do have to know which one uses GTP and which one uses ATP. Yeah, no problem. Okay, so we make IMP following those multiple steps. Sarah, do we need to know the structures? No, you don't need to know the structures. You just have to understand, the components of it. You've got your phosphate, you've got your ribose, and then you've got your base. That's it. Just know the names, essentially. Okay, so here you got your IMP, which is made following the multiple steps. In order to make IMP, you do need ATP, so kind of have an idea as to the importance of ATP in making IMP. Then from there, you can either make GMP or you can make AMP. So the difference here is that first you're going to make these middle products. So you've got your XMP and then your SAMP that are made from this enzyme and this enzyme. I would know both these enzymes. This one's bolded, but this one she bolds later on because we talk about it again. So I would also memorize this one as well. I wouldn't really worry about NAD and NADH, but you do need to know that in order to make AMP, you use GTP. And in order to make GMP, you use ATP. And then obviously know this one, but I think it's pretty obvious when an enzyme is called something synthetase, it's making the thing that it has in the first name, in this case, GMP. So similar to what we were talking about in like 2 slides ago, you have a glutamine, you take the amino group from it and you make a glutamate to add here, and then you wind up with GMP. So the main thing you have to know is that IMP goes through these 2 enzymes, makes these products, so memorize the names of these products. Know that GTP is used to make AMP and then ATP is used to make GMP, which makes sense because you wouldn't want to use ATP to make more ATP. That doesn't, really get you anywhere. There's a question in the chat. What's the small enzyme at the top left? Oh, IMP dehydrogenase? Yeah, memorize that one. She didn't bold it, but she does bold it later on in a couple slides. Okay, so those last 3 slides, I would say know the big picture and most of the details for those because she likes those pathways. This is just a summary slide kind of going over like the main ideas and reiterating what we just learned. Okay, so I wouldn't worry about the atom numbers. That's too much information. But I would recognize that in order to make purine de novo, you're going to need 3 amino acids for the nitrogens. And they're going to come from the aspartate, glycine, and glutamine. So memorize it because it's going to be different for pyrimidines. So there's a question in the chat. Do we have to know which structures are oxidized or reduced? No, don't worry about that. That's too much chemistry. And, she just kind of wants you to know the enzymes and the products and then having like a just general idea of what's going on. So don't have to know what's oxidized or reduced. Okay. So yeah, know the names of these three amino acids and how they contribute to the nitrogens for the purines. Okay, so this is talking about regulation of the de novo purine synthesis. So what she wanted you to note is that if you have an excess of PRPP, that means you're going to have an excess of the reactants. So if you have more reactants, you're going to want to make more products, right? We talked about that in lecture 1, where you have an increase in reactants, you make more products, it goes in a feed-forward activation. So if she were to ask you if there's a lot of PRPP, what would happen? It would, make more products, feed it forward. So that's the big point here. Okay, so your body likes to use the salvage pathway because it uses less energy and it's just way easier. So 90%, and know this percentage because I think she made a big point of it. So 90% of your purine synthesis goes through the salvage pathway. And so pretty much what the salvage pathway is you either eat or, from some kind of cell turnover, you release all these free bases, and these free bases will then need to be put together to make a nucleotide. So from the pentose 5-phosphate pathway, you're making ribose 5-phosphate, and then as we discussed in the first step of purine synthesis, it eventually becomes PRPP. And then you're ultimately going to add this adenine to the PRPP, which is going to be, so the PRPP is essentially like the sugar group. When you add it together, you're going to get your AMP, your nucleotide, and this is the enzyme that you have to know specifically for adenine. For hypoxanthine and guanine, they both use the same enzyme, so just associate them two as using the same enzyme. So here you have your free base that you get through your diet or whatever, and it's also added to the PRPP to make your nucleotides. So it's the same process, just a different enzyme for both of these, for all three of these, sorry. And that's the important thing to make a note of. This, I would say, is lower yield, but the main point she wanted to drive home was the fact that you get your hypoxanthine after, breakdown from the nucleotides to the nucleosides. So you remove the phosphate group. Then you get your nucleosides, and then ultimately you'll get your free bases. So your free bases, your hypoxanthine and your guanine, will ultimately break down further. She liked this enzyme, so I would maybe remember that one, into xanthine. And know this as the first product of breakdown. So know that xanthine is the first product of purine breakdown. And then ultimately it's the xanthine that's going to break down into uric acid, which she goes into here. So yeah, here's your xanthine oxidase. So you see how it has xanthine dehydrogenase here. Don't worry about that at all. Really what it is that the xanthine oxidase is, it's being oxidized and reduced as it goes through this. So it just becomes one becomes the other forwards and backwards as you go along. So don't worry about that. Just remember xanthine oxidase as important for converting both hypoxanthine into xanthine, and then again for xanthine to turn into uric acid so that it could be secreted through your kidneys. And really here she just wants you to understand that uric acid is the end product for humans. Don't worry about anything else, just humans. Okay, so here's your summary. So kind of read through this and get the main points because these are important and likely what she's going to test on. So essentially here you have to memorize these 3 amino acids that contribute nitrogens for purine synthesis. Know that PRPP is the sugar group that is added to the purines for them to become nucleotides. Know your first committed step and the enzymes, the reactants, and the products that go into that. Know that an excess of PRPP can activate or feed forward the reaction to make more product. And that IMP, AMP, and GMP can go back and inhibit PRP aminotransferase to stop the pathway from going forward and making more products. So if these like build up essentially. And then here it's just saying pretty much the same thing, that you got your feedback inhibitions for the first step, and then just read through this and memorize it because these are the main points. And then lastly, you have ATP that needs GM, or ATP is used for GMP, but GTP is used for AMP, so it's reversed. So know that, that's important. And this is just a summary of the salvage pathway. You have your free purine base, and then you add it to your PRPP, which supplies the ribose group, or your sugar group, and understanding that they're catalyzed by these two enzymes, depending on whether it's an adenine, a hypoxanthine, or a whatever the last one was. So yeah, and then understanding that this is 90% of your purine nucleotide synthesis, because it takes too much work to synthesize it from de novo. And then lastly, know that xanthine oxidase is used to convert xanthine to uric acid for excretion. So here she's just introducing these diseases that are associated with purine metabolism. So know that gout and Lesch-Nyhan syndrome are both associated with purine metabolism. So essentially what happens here is, for whatever reason, because your kidneys aren't working properly or whatever, you're going to have an increase in uric acid in your blood. And what happens is that it can't dissolve in your blood after the concentration passes a certain amount, similar to when you try to make rock candy and it starts to crystallize. It will crystallize in your joints. And it also can crystallize in your kidneys, which is trying to filter out the uric acid, but is unsuccessful. And so, you can also have crystallization in your kidneys, which can cause kidney stones. So, yeah, and so then you get these inflammatory processes. So the uric acid builds up in your joints, and then your body's inflammatory cells will come in and try to get rid of it, but obviously that just makes everything painful, swollen, and uncomfortable. And then this, don't worry about it. She just kind of mentioned that you mostly get the symptoms in your fingers and toes, and she was just showing you pictures. Okay, what you do need to know is that in order to fix this problem, you take a drug known as allopurinol that you have to know, and it's going to inhibit xanthine oxidase. So when you inhibit xanthine oxidase, hypoxanthine cannot be converted to xanthine, and then xanthine cannot be converted to uric acid either. So then what you wind up is with an increased concentration of hypoxanthine, and she'll show it here. So the allopurinol works because it looks structurally similar to hypoxanthine. So if you have all of this allopurinol hanging out in your bloodstream, it's going to be more likely to react with the xanthine oxidase and essentially flood the enzyme so the hypoxanthine can't use it. So then you have hypoxanthine that is more soluble in your blood and can be secreted easier. And that's how that drug works. So kind of understand how it works as well. Because I think she put it in her summary slide at the end. I would also memorize these drugs because she did bold them. But essentially, the probenecid, it decreases the renal reabsorption. So when you decrease, Fernando, what did you want me to repeat specifically?
The last slide when you talked about urinal, you said that the secreted part, the part that says that it's easily secreted.
Oh, okay. So yeah, hypoxanthine and xanthine are more soluble in your blood. So if you just have higher concentrations of those because they're not being converted into uric acid, they're able to be secreted from through your kidneys easier than uric acid would, which would, at higher concentrations tends to crystallize. Does that make sense?
Okay, thank you.
You're welcome. Okay, for this, like I was saying, if you decrease the reabsorption of urate, so uric acid, you're going to secrete more uric acid into your, urine, essentially. So you're getting rid of it from the blood and peeing it out. And then for these, this is just for symptom relief. So when you reduce inflammation, your body's not going to respond as dramatically to the uric acid crystals in your joints, and it just makes it less swollen and less painful, essentially. So memorize these and associate them with gout. Okay, for Lesch-Nyhan syndrome, here you have a deficiency of hypoxanthine guanine phosphorybotransferase, so HGPRT. And remember that this is the one that converts hypoxanthine and guanine into IMP and GMP. So if you don't have this enzyme, you're unable to make IMP or GMP, or you're not able to make enough of it. So that winds up with hypoxanthine increasing in concentration because it's not being converted into IMP. And so as a result, you have hypoxanthine going down the uric acid pathway where it turns into xanthine and then down to uric acid. And you get the same problem where you have an increased amount of uric acid. Know that this is linked to your X chromosome, and therefore, because boys only have one X chromosome, you'll see it more often in boys. And kind of memorize some of these symptoms in case she uses it in the test for you to identify this. But I would say the sand-like uric acid crystals is probably one of the more important symptoms to remember, as well as like the jerking of the muscles and all that. Okay. And so understand that LNS can also be treated with allopurinol to decrease your uric acid, but it's not going to help the other, neurological and behavioral symptoms. So it's just good for reducing kidney stones, essentially, in people who have this. Okay, so pyrimidine metabolism. So same thing, it has its own de novo and salvage pathway, and it also has its own catabolism mechanism. So this is a very important slide to remember because remember, she likes the first step and then the committed step. So understanding that glutamine, carbon dioxide, and ATP will come together through CPS and then make carbamoyl phosphate. That's what you have to know from this slide. And that's the first step. Know that that's the first step. Then you have to know the committed step. And you have to know that it's going to act through ACTase. She likes this abbreviation, if I remember correctly. Know that your carbamoyl phosphate that you made in the first step is going to be combined with now aspartate. So memorize that. And they're going to go through ACTase to make N-carbamoyl aspartate. So that's probably the most important part, but I would also memorize dihydroorotate to make dihydroorotate, because that's really easy to remember since it's got like a different ending, but it's kind of the same thing. And then your dihydroorotate dehydrogenase to make your orotate or orotic acid. So I would definitely memorize the enzymes here. They're not too difficult because they're kind of very similar in name to the products. But this is definitely an important slide to remember, and know that this is the committed step with this particular enzyme. So Lindsay, how would she word a question like this, though? Which step is the committed step, or would she give us a diagram? So she wouldn't give you a diagram. She probably would ask you, not like as directly as which is the committed step, but she would maybe put it into like a vignette or something like that, and kind of tell you like to say the enzyme or whatever is the product or reactants. So hopefully that helps. But yeah, she kind of does really like you to tell her what the enzyme is. Okay. So this is also super important to know, is the bifunctional enzyme. So what they mean by bifunctional enzyme is you have a protein, and every protein has an N-terminus and then a bunch of amino acids, and then you got your C-terminus. So what it means is that the name of this enzyme, this is the name of the whole thing, is uridine 5 monophosphate synthase, but it has its N arm and then it's got its C arm. And the N arm, you have to know, is this. The name of it is this, and then the name of the C arm is OMP decarboxylase. So here you have your orotate, which you made in the last step. You're going to now add your PRPP from the ribose 5-phosphate, and that's going to contribute your sugar group. So note that with pyrimidine synthesis, unlike purine synthesis, your PRPP is going to be added way later on, unlike purine, which has it as the first step. And this is important to differentiate between the two of them. So your N-terminus is going to be known as OPRT, or this long word, but I think she likes the abbreviations for this. So either way, whatever makes you comfortable. And then it will make OMP. And then after that, the C-terminus will act on it as OMP decarboxylase, and it will make UMP. And from UMP, you can add ATP to make UTP, but the main point here is this is how you make UMP. So this is just another summary slide. She's kind of outlined the most important enzymes. I've kind of made a point to show you where the N-terminal enzyme and the C-terminal enzyme occur, so you can like see that a little bit better. But yeah, I would probably memorize the names of both the N-terminal and the C-terminal enzyme along with the name of the all-encompassing enzyme that includes the two of them. Okay, Jessica's asking, can you repeat what you said about the PRPP being added for purines and pyrimidines? Yeah, so a big differentiating factor between purine and pyrimidine synthesis is the fact that with purines, PRPP is the first step, meaning that the base is being kind of built on the purine, on the PRPP. Whereas with pyrimidines, the base is going to be created first, and then it's going to be put on top of the PRPP later on, because you don't see it until like what, step 5? Whereas in purine synthesis, it's the first step. Okay, so this is just showing you that you can convert UTP into CTP. I honestly wouldn't remember this enzyme because it's pretty obvious, and she's not going to ask you what does CTP synthetase do because it's literally in the name. But I would understand that UTP can be converted into CTP. This is a little bit more important to understand because this is how you go from like an RNA base to a DNA base, because now you're doing the deoxynucleotide versus the ribonucleotide. So there are two enzymes that kind of work together to do this. You've got your ribonucleotide reductase, which is the main enzyme that allows a nucleotide to become a deoxynucleotide. And then it kind of works alongside these redox enzymes. So what these redox enzymes do is they have these disulfide bridges on their proteins, and then they use NADPH and take the hydrogens from the NADPH and break their disulfide bridges. So now they can give these hydrogens to ribonucleotide reductase for it to add it right here and, catalyze whatever reaction it has to do in order to add the hydrogen. So the redox enzymes provide the hydrogen, but the ribonucleotide reductase is what's doing all the work. So know these two enzymes, understand that it uses disulfide bridges to do this, and NADPH helps as well. But this is really just telling you that you can go from RNA to DNA. Okay, so this is the same concept. Thymidylate synthase is not something I would remember because, like I said, it's pretty obvious what it does, and she's not going to ask something so obvious. But just understand that you can take uracil and turn it into a thymidine. So if you were to convert something from, say, RNA to DNA, you would first make it a deoxy. So this is why you have this really weird nucleotide that you would never find in neither DNA or RNA, but it's just kind of like this middleman um, who exists until you can turn it into a thymidine. Okay, there's a question in the chat. Are Dr. Patel's questions and Dr. Ferreira's questions going to be combined? Oh, I see. So no, what you learn in Dr. Patel's lectures, it's through like based on each lecture. So you're not going to see like any questions about disulfide bridges specifically in, say, this this lecture. So they're not really going to combine them, but like it's important to understand Ferreira's lectures to better understand Patel's lectures. But Patel's not going to ask you anything that's, based off of like lectures 1 or 2. I mean, 2 or 3. Does that answer your question, Catherine? Okay, cool. Okay, so yeah, so you get this like middleman here that only exists for a short amount of time in order to become a DNA base, and in which case it would become thymidine. Okay, so here is what you need for the nitrogens for pyrimidine. Since pyrimidine is only going to be 1 ring, you're not going to need as many nitrogens. So that's why only 2 amino acids play a role in supplying the nitrogen. So know what these 2 amino acids are and be able to compare them to purine synthesis. So while purines need aspartate, glycine, and glutamine, pyrimidines only need glutamine and aspartate. So they're missing the glycine. They don't need the glycine. So know that. And then this is what I was talking about earlier, how in the purine ring, you have the first step being PRPP, which allows the purine ring to be built on top of the PRPP because it's the first step. And remember, the PRPP is what supplies the sugar group for the nucleotide. However, with the pyrimidine ring, because PRPP is added way later on, your pyrimidine ring is going to be pre-formed, so it's going to be made first and then added onto the PRPP. So know this difference between purine and pyrimidine synthesis, because this is kind of, I think she asked a question on it, to be honest. Okay, so this is just the same thing that we've seen before, but it's with pyrimidine, so this is how you would regulate it. So the main thing that I would remember is that the product, CTP, can go back and inhibit CPS, which is the first step, and inhibit its own production. So this is just regular negative feedback loop, where a lot of product builds up, it goes back and inhibits the enzyme that makes it. And then additionally, did somebody have a question?
No, I was going to say that in the lecture, she said something about ACTAs that I don't know if you were going to get into that, but I was kind of confused about that.
I don't remember what she said exactly about that.
Getting the CPS, but then something about also about the ACTAs. So that's why I was like, okay, but where is that in the diagram?
Oh, I guess I see what you're asking. Okay, so maybe she was talking about like CPS is the first step and then ACTAs is the committed step. Maybe she was asking you to differentiate between the two. So ACTAs is the second step. So it wouldn't be this step. It would be the second step. Can you guys see my pointer, by the way?
It's a little different.
Very small.
Oh, yeah, I'm sorry. That's so weird.
So unless you're like writing something, it's kind of hard to like follow it.
Okay, I'm sorry. I didn't even realize that. I'm like looking at my other screen now and like, yeah, I don't even see it. Okay, so I'll just like circle everything. Okay, so essentially you have your CTP goes back and inhibits CPS, and it stops CPS from going down this whole pathway. PRPP, remember this comes in later. It is going to be made by, the first step in purine synthesis. So that's where it comes from. But it's just kind of floating around in the cell, and if it comes in contact with CPS, it will upregulate it. Because remember, if you have plenty of PRPP floating around in the cell, you're going to want to make more product. So that's what she was getting at here. Same thing here, ATP, if you have a lot of it, you can make more CTP. So same kind of general concept. If you have a lot of reactants, you can make more products and it's going to be pushed in the forward direction. And then, I mean, these are lower yield because like it's kind of obvious, but knowing that UMP inhibits its own production and that CTP inhibits its own production directly. And these are the enzymes that are doing this. Okay, so this is a lot of information, but a lot of these enzymes, like this one in particular, we've seen before. So we know that ribonucleotide reductase, it's the one that works with the two redox enzymes to go from an RNA nucleotide to a DNA or a deoxynucleotide. So that would be your first step, is making it a deoxy. Hence why you'll make this deoxy UDP, which is not something you'd find in DNA. And then from there, you're going to have a kinase. I don't think it's super important to memorize the name of this kinase. Just understand that it's a kinase, which then adds a phosphate to make it a 3-phosphate group, so a ATP in this example. And that's what you would use for DNA. It gets tricky when you start talking about uracil, but if you remove the phosphate, you would then make it a DUMP. So understand that it's DUMP that needs to be created first in order for it to be converted into DTMP, like we saw in one of the last slides. So this is that same enzyme, which pretty much tells you what's being made. So it's not super important to remember because it's so obvious, but you still might want to understand that that's the enzyme. It's also important to note that you can make a CMP, DCMP, and make it into a uracil, which we already kind of talked about as well. And I don't remember if we have to remember this enzyme, but I probably would just in case. So know that, and then you can complete the pathway to make it into DNA. So the main point of this slide is that she wants to show you how RNA bases are turned into DNA bases. Okay, so now we'll start talking about the pyrimidine synthesis for the salvage pathway. So here you'll have your free bases that you get from your diet or whatever. And you'll have either uracil or cytosine because they go both through the same exact enzymes. So first they'll go through this pyrimidine nucleoside phosphorylase. So remember this because she did type it up and kind of made it blue so you could see it. So it will act right here, and it will also act for the thymine as well. So they share the same enzymes between uracil, cytosine, and thymine for the first step. However, with thymine, because you only see that in DNA, you're going to use the deoxyribose to add on the sugar group until you get your nucleosides. So these are your nucleosides. They have your sugar group, and they have your free base attached together. So the next step is to turn them into nucleotides, in which case you would use thymidine kinase to make it a TMP, so you have your phosphate group added, or uridine kinase, also known as uridine cytidine kinase. It's the same enzyme, just two names for it. I would probably just remember one and kind of figure that. You can figure it out if you see the other name on the test. And then that will, as a kinase, will phosphorylate it and turn it into UMP, which is now a nucleotide because the phosphate group has been added. So I would memorize this pathway, but it's not too difficult. So hopefully there's no problems there. And this is more of like a big picture idea here. She wants to show you pyrimidine catabolism, but she's not going to give you all of the enzymes for it because that's too much information. However, she's just kind of giving you that, you have your RNA, and then all of these yellow highlighted ones are going to be your nucleotides, and then your nucleotides will ultimately break down into nucleosides. And then from nucleosides, you break them down into two types, your uracil and your thymine. So know that these are the two final free bases that they break down to when they do break down. Once again, you know that your cytosine can be turned into a uracil. So this is the same concept here. And that's in order for it to break down as well, you need to turn it into deoxyuridine. But other than that, just understanding the general idea that it goes from nucleotides to nucleosides and then ultimately makes up uracil and thymine as the two free bases. Because then she wants you to understand that you go from uracil and thymine, and they break down into two final end products. So know these two end products as the end products of catabolism for pyrimidines. So you've got your beta-alanine and your beta-aminoisobutyrate. And she said not to worry about any of this, so I wouldn't worry about it. So just pretty much everything in this box is all you have to know for this. So this is just another summary like before. Make sure to read it because it's got the big points to it. Gabrielle asked a question. Do we have a Q&A session tomorrow because of the short week? Thanks. Yes, we do have a Q&A session tomorrow. It's going to be in person. And we also have a quiz for you guys tomorrow as well. It's the same deal. It opens up at 11:15, closes at 1:45. And we have fewer questions this time, so it's only 55 minutes long and will be like 36 questions because Ivy and I only contributed one lecture each this week, so we only contributed 6 questions for our lectures. So it's 36 questions. And it's in person for Group A. So if you're Group A, you show up at the auditorium. If you're Group B, you get to show up next Friday, and then it like alternates between the two. But it's also being streamed on Teams. So for anyone who's online or is Group B, you'll watch it on Teams tomorrow at 2. Okay, moving on. So the other thing to remember that we already talked about is that the pyrimidine ring is preformed and then transferred to PRPP. Unlike the purine ring, which is built on PRPP, you need to know the first committed step and know the enzyme for it as well as the product. Also know the feed-forward activation with same thing as purines here is the PRPP increases and you wind up with more product because of that. And then knowing that you can inhibit CTP at CPS to stop the production of CTP. Okay, and then same thing for the salvage pathways, understanding that you take a free base, and then you take the ribose 1-phosphate, and then you add them together by pyrimidine nucleoside phosphorylases. And then again, with the 90% of your pyrimidines, like the 90% of your purines, they're both going to be through the salvage pathway. And then for catabolism, like I said, the most important thing to understand here is that beta-alanine and beta-aminoisobutyrate are going to be your final end products. She also added these as your end products as well, but I would say those are lower yield. Okay, and lastly, she's going to be talking about pyrimidine metabolic disorder. So you only have one pyrimidine metabolic disorder, and it's this. I would memorize both names because either or could show up on the exam. So know both names. So you should also be able to understand that this is essentially a dysfunction in the bifunctional enzyme that we talked about. Remember, it's the one that has the N group and then the carboxyl group, the N terminus and the C terminus. And so if you have a dysfunction here, you're going to have issues converting orotate to OMP and then converting OMP to UMP. I would probably memorize the symptoms associated here. So because you have an issue making UMP and OMP, you're going to have an increase in orotate. So that's orotic acid is going to accumulate, and like uric acid, accumulation of this stuff is not good. However, unlike gout, you will have to treat it with Zuridan, and just memorize the name of this treatment. Okay, so for this, don't know any of it. She was just adding that for you in case you were curious about it. But yeah, and then read the summary slide to review the material. Okay, that's the end of our one lecture. I did make a Kahoot that we can play, but I can answer questions first. So Sarah, so the quiz is not cumulative, not 3 weeks worth of material, only this week's material. Yes, the quiz will only cover what we learned that week. So it's not cumulative at all. Does anyone else have any questions about that before I start the Kahoot?
I actually do. If you're in section A, does that mean that you have to go to the in-person Q&A?
No.
Okay.
Yeah, no, you don't have to go if you don't want to.
So you can still watch it online, right?
Yes.
Okay.
Yeah, of course. Just to confirm, the quiz is at 11 and then the Q&A is live at 2. Yes, the quiz opens, I think, at 11:15 and then the Q&A is at 2 p.m. Yes. Can you please go over the bifunctional enzyme again? Fernando, what part of that did you want me to go over? The mechanism, or did you want me to go over the disease?
Just the enzymes in general that you said that were, because you said that one was C-terminal based, the other one was N-terminal based, and then yeah, something along those lines. That it was like pretty fast, so I didn't.
Oh, okay, yeah. Hang on, let me go back to where we talked about that. I think it was here. Okay. I don't know if you can you see my cursor okay?
Yeah.
Okay. So this is the name of the enzyme as a whole. And then it has an N-terminus. So you know how a protein has an N-terminus and then it has the end of its polypeptide chain is the C-terminus. So at the N-terminus, it's going to be called orotate phosphoribosyl transferase, or OPRT, and that's going to do the first step to make OMP. Then the C-terminus of that exact same uridine 5-monophosphate synthase its C-terminus is known as OMP decarboxylase, so it's going to make UMP. So the same enzyme has two parts of it that is working to make this step and then this step. Does that answer your question?
Yeah, thank you. Sorry, it was just confusing.
Yeah, no worries. It's totally confusing when you're first going over it. Does anyone have any more questions, or can I start the Kahoot? Okay. Well, I'll start the Kahoot, and if anybody has any questions, you can ask after. So it's only 5 questions this week. I'll give it about 1 more minute before I start, just in case anyone's having issues getting in. Okay, it looks like it's stagnant at 38-ish. So I'm going to go ahead and start it if that's okay. Awesome, great guys. Yes, remember that adenine, xanthine, and hypoxanthine are the purines, and then uracil is a pyrimidine. Right, awesome. Yes, arginine is not one of the 3 amino acids that contribute nitrogens to the purine rings. It's aspartate, glutamine, and glycine that do. Awesome. Oh, kind of. But yeah, remember that CPS is going to be the first step in pyrimidine synthesis, and ACTase is going to be the second step, aka the committed step. So make sure to memorize the difference between the two. Yep. Just another memorization question. It's the 2 betas that are going to be the end products of the pyrimidine catabolism. Awesome. Great, guys. So the red and the orange answer choices are the 2 names for the same thing. It's the pyrimidine metabolism disorder. And then obviously it's not gout. But yeah, Lesch-Nyhan syndrome is the one for that. Just have to memorize that stuff. All right. So who won? Nice job, Brandon. Lindsay, to answer your question, I did post the link to the first Kahoot. It's the last slide on, I think, the globular proteins lecture. So I made like a little link to it at the end of those slides that I posted. But I can post the link to all of them as like a separate thing if it makes it easier. I still haven't posted the link to this one, but I'm going to do that tonight. So if anyone doesn't have any more questions, you guys are free to leave, but stick around if you do. You're welcome, guys.